All right, everyone. What is up? How's it going, Titan? Man, I'm trying. I got this the chat set up, but it refuses to show up. I don't understand. It should show the YouTube and Twitch chats, and it's not. It's very frustrating. I can't troubleshoot it, so I'm just gonna kind of go with it. Let's see. Just gonna double check and make sure everything's being recorded all right. Hope everything is going on your end. Hey, Ghosts. Hey, Titan. All right. Output, recording. What? All right, let's try this out. Oh, yeah, I'm trying out livepush.io. It's like this thing for I used to use Restream because when I was doing Twitch and YouTube, but I've kind of abandoned it just because it's like $20 a month. They went from free to $20 a month. So I am like, okay, that's way too much. That is way too much. Yeah. Let's see here. Just want to make sure the audio is coming through real quick. Perfect. Cool. Oh, that's nice. That's nice of you, man. Cool. So I got like a whole list of things I want to start. This is just like going to be a recording stream. I want to try to do this more regularly, kind of just go through the things that I've seen that are kind of cool. All right. It's going to be kind of annoying. Okay, 1 second. All right. I think this is it, though. Oh, what's up, Nick? How's it going, man? Thanks for coming. This is nice to see you. So, we got some cool... I mean, I was going to go over papers and stuff. What were you thinking? Are you streaming right now, too? Let's see. I'm down, though, if you want to. That's fine. All right. This should work. And then... Cool. All right. I don't know why the multi-chat's not working, but you're in YouTube chat probably. So I don't know. I just felt like I might as well throw the Twitch in there as well. I don't have anything to lose, you know. All right. Yeah, I'm just using their browser, and then I'm going and accessing it, and then it should be like showing up here, but it's not. It has the not counting like any destinations, but I'm logged in, and it's streaming to the it's streaming. So that's what I don't understand. It should be it should be going. All right. Anyway, it doesn't matter. It is what it is. Okay, cool. Let's do this. All right. Cool. Oh, thanks, Nick. Yeah. What do you do? What do you use for cross-platform? Like, I'm curious. Restream is $20 a month, man. I can't afford that. That's ridiculous. Just for restreaming, I'm not like. That's like, Live Push does free, I guess, or maybe they don't, and that's the reason why the chat doesn't work. But it's restreaming right now to my Twitch and my YouTube, so I think that's kind of cool. I wonder if we could just go to this. But it should be oh, you know what it was? I don't want the restream ad. It's like a big watermark on it. So I'm like, what's up, Trusty Ape? So I'm like, you know what? I don't like the branding. I don't like they're not paying me. You know what I mean? If you're putting my stuff. Yeah, it's the brand. It's the watermark that I'm kind of like over. So okay. All right, everyone. We got a really cool paper, kind of like the convergence of AI and biology. What can AI actually do in terms of creating new ways that we see drugs, that we can use them for biopharmaceuticals? Basically, AlphaFold thinks that they kind of have pretty much everything figured out already, predicts the shape of nearly every known protein. And this is going to be kind of something that has been used a lot. If you remember folding at home and stuff like that, the idea has been to actually try to use the ability of multiple computers and computers all across the world to basically figure out these intimate folding between proteins and of proteins themselves. So the idea is to kind of use these AI tools to actually figure out the structures, and then maybe we could find new antibiotics, new ways to actually interact with binding proteins in bacteria or whatever, and that would be an antibiotic, right? So here's the paper. This is just a Science News article. So this is the structure of a vitelligen protein, a precursor to an egg yolk, as predicted by the AlphaFold tool. So from today, determining the 3D shape of almost any protein known to science will be as simple as typing in a Google search. So we kind of have like similar things right now. There's something called UniProt. So UniProt is kind of like what we have right now. So like, let's say I needed to find like the shape of Rubisco, for example, or something that's a really popular thing. So let's go to the cards, and then now we can actually kind of see by organism, Rubisco by human. That's that's a little weird. Rubisco is the molecule that is actually the protein, I should say, brings down the carbon dioxide into photosynthesis. So it's a really well-studied protein. And so let's look at Thaliana, Arabidopsis. And this is kind of like what you'd be looking at currently. So this is kind of our current structure. You kind of have all the amino acids, you have the protein descriptions, where it actually is in the cell, it's in the chloroplast, what pathway it's in, and this is kind of like what we normally do in science if we're looking for information. And already you can see, like, biochemically, the ones that we've studied are really well characterized. Rubisco being one of the ones that we have the most science behind it. And this is kind of like the 3D structure here. This is actually really important because you can actually figure out the binding sites or the active sites, or basically the parts of the protein that actually do the work. So if you think about, like, your hand, the active sites are kind of like your fingers, the parts of the protein that are actually going to interact with stuff, and how things actually interact in the real world are going to be a lot different than what are kind of modeled. So my prediction would be that this, there's a lot of promise in these new technologies, but the problem kind of is that how are they actually going to perform? So normally what they would do would be to have some predictive capabilities and then some testing. So I'm curious, like, how are they going to build out that knowledge beforehand in order to be, like, super, super useful? Because generating a bunch of proteins that aren't going to be doing anything useful, well, isn't that useful, right? So let's think about that while we go here. So researchers have used AlphaFold, the revolutionary AI network, to predict the structures of more than 200 million proteins from some 1 million species, covering almost every known protein on the planet. That's a lot. So humans, for example, I think that there's like 100,000 proteins or something that make up a human. So that is a lot of proteins. The data dump is freely available on a data set set up by DeepMind. That's really good. We want to make sure that the data is freely available so that we can actually access it and use it. Essentially, you can think of covering the entire protein universe. The DeepMind chief executive Demis Hassabis said at a press briefing, "We're at the beginning of a new era in digital biology. The 3D shape or the structure of the protein is what determines its function in cells. Most drugs are designed using structural information, and the creation of accurate maps of protein amino acid arrangements is often the first step in making discoveries about how proteins work." Yeah, kind of like what I was saying before. We have a structure, and then figuring out which part of that structure is going to kind of do the work is what the whole point of putting this into practice. A lot of times we can actually mutate parts of the genetic code or the DNA in order to change those binding sites and then kind of see what those mutations are going to do. So this is kind of like what they're talking about there. So DeepMind developed the AlphaFold network using AI techniques called deep learning, and the AlphaFold database was launched a year ago with more than 350,000 structure predictions covering nearly every protein made by humans, mice, and 19 other widely studied organisms. Wow. Yeah, because I wonder if they used UniProt, which kind of like what we were looking at before, just because what would be how could they learn? So this is deep learning. There's a couple of different AI models, but deep learning they would have to give them a lot of structure predictions already, and then I'll kind of use that knowledge to then you could go forward and create other foldings that exist. The catalog has since swelled to over 1 million entries. That's good. We're bracing ourselves for the release of this huge treasure trove, said Orenjo, a computational biologist at University College London, who has used the AlphaFold database to identify new families of proteins. Having all the data predicted for us is just fantastic. Yeah, if you can have like results that are easily used and stuff that can kind of be used with easily is the best way to go about it. You don't want to have to redo all the data every time that you'd want to do this. Okay. So the release of the AlphaFold last year made a splash in the life sciences community, whose members have since been scrambling to take advantage of the tool. The network produces highly accurate predictions of many protein structures. It also provides information about the accuracy of its predictions, so researchers know whether they can be relied on. Conventionally, scientists have used time-consuming and costly experimental methods of X-ray crystallography and cryo electron microscopy to solve protein structures. Yeah, there was a time in biology or any, honestly, like biochemistry, I guess more broadly, where you could basically do X-ray crystallography of like any protein and almost get a Nobel. It's an exaggeration, but you look at the last 50 years of Nobels and 40, 50 years ago, just getting the structure of the protein was enough in order to such an advancement in science. Through X-ray crystallography, which is basically like freezing the protein and then kind of like looking at it like with shadows and stuff. I don't really know it entirely myself, but the idea is you're getting basically the structure, and that in and of itself was enough to get Nobel Prizes. So this is a huge step forward in order to get all this information. Very cool. So according to EMBL EBI, around 35% of more than 214 million predictions are deemed to be highly accurate, which is what we were talking about before, which means they're good as experimentally determined structures. Another 45% are considered to be accurate enough for many applications. Interesting. I mean, what you would do there, so you'd have all these all these different predictions, right? So how do you actually test it? So you would do some type of like it's called like multiplex high throughput assay, so that you have you're actually changing nothing in the experiment, but you're actually the molecular work and the biochemistry is actually doing the hard work of figuring out which protein actually does it the best. And then maybe that protein structure is turning on a fluorescent protein or something that you can tell through microscopy, like, oh, this actually worked. And then you could look at, okay, so if this turned on and this protein made that turn on, what is that protein structure? And then you have kind of like a unique tag or something on the proteins. So then you could actually do this through like liquid handling robots, that kind of stuff, to really see the efficacy of each of these mutants, which is, they need to be tested. So many AlphaFold structures are good enough to replace experimental structures for some applications. In other cases, researchers use AlphaFold predictions to validate and make sense of experimental data. Poor predictions are often obvious, and some of them are caused by intrinsic disorder in the protein itself. That means it has no well-defined shape, at least not without other molecules present. The 200 million predictions released today are based on the sequences in another database called UniProt. Oh, that's so funny. I didn't pre-read this, but yeah, they used that. I mean, that's the database that everyone kind of uses in science, so I'm not surprised, It's likely that the scientists will have already had an idea about the shapes of some of the proteins because they are included in the databases, experimental structures, or resemble other proteins in their repositories. Cool. Well, I think it's just like another way of figuring out all this stuff, how we're going to interface these new technologies and these new capabilities that we're having with computers to leverage that into new ways. So there's been a lot of development in here. So now we kind of need to apply it, and applying some of my favorite papers have actually been applying machine learning or deep learning techniques with actual like evolution based, so directed evolution, and that's actually using evolution to select for the best mutations, and then you kind of use that and then mutagenize the winners and then increasingly mutagenize and make them better, testing each iteration as you go forward, kind of like a hyper speed evolution trail that will create products really quickly. If you're interested in that, Frances Arnold, one of my favorite scientists right now. She has an incredible work with directed evolution. I think she got the Nobel in 2017 for 2017 for directed evolution. But just incredible stuff. Really, really cool. So, all right, cool. Yeah, some of the mutations are just bad. They're just going to be bad. They're going to have errors in them, folds that are just don't make any sense. That kind of, they just have no use whatsoever. All right, let's do. Dang, I want to watch a TED video, but last time I did this, I got copyright struck. I was pausing it and like watching it and pausing and watching, but it wasn't good enough. And I got my stuff, my channel taken down, and I'm like, it wasn't like a strike or anything, but it was a copyright infringement, whatever, that they actually shut down the stream. So I don't know what to do. I really want to listen to this. I don't know if it's worth it. So stupid. I mean, there's people stealing entire videos from people, you know, it's like, it's ridiculous. There's that if you wanted to look at it. Here is the, here's the paper that we were just reading if you're interested in it. I don't think it's worth it. This particular video is about actually using fungi to create new materials. So I think that's like the cool part about it. It's like using and leveraging fungi to actually to make new buildable materials like clothing. This is it. I don't know. I think I'm just gonna maybe skip it. It's weird. It's weird. Like the bigger streamers have more privilege because they're like just going through like, they're just going through full videos, just watching it, you know, maybe reacting to 10 minutes of content. But I watched a microscopy video on YouTube and it got shut down immediately. All right. All right. This is going to be an interesting one. Okay. All right. We're going to read the abstract in here and try to see the figures and then go forward, I think. So this is a cool one. All female salamanders exist and they actually evolve. So this is really cool. So there are all female unisexual salamanders that give birth or make new copies of the salamanders through things called parthenogenesis. So this is the ability for XX chromosomes to actually replicate themselves and actually create a new way of making, instead of doing like meiosis, which would be like the standard kind of way that you'd make a sister gamete and then they would come together. It actually creates two X chromosomes that cross each other. And it's a really insanely cool process. But how do they actually introduce new DNA is a really good question, right? How are they not just driven to extinction because no new DNA are kind of introduced to the system, therefore, and how do they not just kind of die really quickly? So this is a really cool example. This all-female lizards or salamanders, salamanders. So let's check out this paper. Bogart et al. Bogart, Berzofsky, Nobel, and B. 2009. Although bisexual reproduction has considerable evolutionary benefits, several all-female vertebrates exist. So this is basically things that have gone through parthenogenesis and have only all-female copies, only all XX in their species anymore. So this has happened in sharks, which is really interesting. There was a shark that was born in a tank hadn't seen a male of the species in like 10 years or something. And they're like, there's no way that it would have happened. And then they did the genetics of it. And sure enough, it was parthenogenesis. So really cool. Unisexual salamanders in the genus ambystoma are common around the Great Lakes region in eastern North America. They originated from a hybridization event that involved a female that shared a common ancestor with Ambystoma barbouri 2.4 to 3.9 million years ago. But unexpectedly, A. barbouri nuclear genomes were unknown in unisexuals. Wow, interesting. Were unknown in unisexuals. So basically, they hadn't found this nuclear genome in the other one. They didn't know that it actually had the only like bisexual reproduction would have worked. So unisexual salamanders steal sperm from donors or normally bisexual species. So their reproductive mode is described as kleptogenesis. Whoa, I thought it was parthenogenesis. Wow, what? This is nuts. Wow, I didn't know. Wait, what? I am just learning about this. This is incredible. I didn't know that this was possible. So they steal sperm? But they're all females, so they have to have, where does the Y chromosome? The Y chromosome can't come from that sexual reproduction. So is it just, where does the Y chromosome go? Unisexual salamanders steal sperm from donors of normally bisexual species. So their reproductive mode is described as kleptogenesis. Wow. Most unisexuals are polyploid, and they all possess at least one alateral genome. So polyploid means that there's multiple copies of their genome, and they all possess at least one alateral genome. Wow. Wow, that means that it's such a diverse way of having their genome stored. One or more other genomes are taken from sperm donors that may include A. jeffersonium, A. laterale, A. texicum, and A. tigrinum. We examined unisexual adults and larvae in a southern Ohio pond where unisexual individuals coexist with male A. barbouri. Whoa, this is really cool. So I would be thinking about like comparing, I would think about comparing the genomes of them and see if they actually aligned. But then also it'd be really cool to actually see where it goes. Like I would want to tag the DNA or figure out if we could like actually follow it physically, just actually see what is actually happening here. Let's see if they do that. So this population provided an opportunity to test hypotheses pertaining to the role of A. barbouri in the evolution of the disparate cytoplasmic and nuclear genomes in unisexual salamanders. Okay. Microsatellite DNA loci, so mitochondrial DNA sequences and genomic in situ hybridization were used to identify the genomic constitution of the individual. So these are the different ways. These are the different ways now that they're actually trying to show where the DNA is going. So they're using, they're actually using some type of marker, mitochondrial DNA sequences. So this is kind of interesting, mitochondria, because they're, endosymbiotic theory, they're actually been engulfed and they used to be their own, proteoarchaea or bacteria. So mitochondria in your cells, you actually have two genomes as a human. You have your human genome, which is in your nucleus, and then you have your mitochondrial DNA genome, which is in your mitochondria. And this is kind of cool because they actually have different methods of transmission. So like, for example, so in humans, you get like 99.999% of your mitochondrial genome from your mother, because basically the egg and the sperm, as they meet one another, the sperm, all the mitochondria for the sperm, think about it, like where it's only just moving, right? That's the kind of like the whole goal. So there, all the need for any type of machinery that's going to create like ATP and all that kind of stuff is all in the tail, right? It's just bringing that thing going. So when the sperm meets the egg and that tail actually detaches, you actually get the mitochondrial leaf, the sperm. So the sperm head buries into the egg, but the sperm at that point, because it's left its tail behind that has all the mitochondria, the subsequent zygote has just mitochondrial genes, DNA, just from that big egg. The big egg has like a ton of resources, right, that need to maintain the DNA. It needs to go through a lot of different cycles, so that has a ton of mitochondria. So it's basically outpaced by a ton. So that's why basically you can do mitochondrial DNA sequencing, and that would go through the female lineage of your past. Very cool. And people know this from when you try to sequence your own genome at many of those 23andMe places or whatever. You can actually do mitochondrial DNA sequencing separately, which is kind of cool. And genomic in situ hybridization. So genomic in situ hybridization is a way, this is familiar to fluorescence in situ hybridization. So that's going to be a way to actually see where the genome is, which I'm really excited about, because we're actually going to be able to see where this genome where it goes through microscopy. Okay, so they were used to identify the constitution of the individuals. A. barbori was found to be an acceptable sperm donor for unisexuals, but only contributed genomes in ploidy elevated individuals. Wow. A. barbori was found to be an acceptable sperm donor for unisexuals, but only contributed genes to the ploidy elevated individuals. So ones that didn't have any type of increase in the amount of genome size didn't have any of this DNA, which is interesting. In the absence of A. jeffersonium, this Ohio population is likely experiencing a recent switch in sperm donors from A. jeffersonia to A. barbori, and demonstrates the evolutionary flexibility and dynamics of kleptogenesis. Oh, that is so cool. All right, let's go to the figures. Let's see what we can actually see here. So these are them. Representative salamanders found in the Mitchell Memorial Forest Pond. That's so crazy. That's so crazy that you could get like all this stuff happening right next to you, and you honestly wouldn't even know that it was happening. You know what I mean? That it's just like they're just there in a random pond, and you just hike past it. You have no idea what's going on. Representative salamanders. There we go. So that's what they actually look like. So A is the figure 1. Which is the top one? Oh, they killed these. They killed them. They've been deposited in the amphibian collection. RIP. Thanks for your contributions to science. So the A. jeffersonium was the top, and the A. lateral barbori was the bottom. Oh, this is the hybrid. Yeah, A. lateral jeffersonium barbori was the middle, and A. barbori, which is the new sperm donor, is on the bottom. Wow. It doesn't have a lot of it doesn't have a lot of the characteristics that you would associate. None of them. None of the phenotypic characteristics. That's weird. Cool. Fluorescence genomic in situ hybridization, GISH. This is before the I guess we're this is, we want to visually see. These are actual chromosomes. Cool. Fluorescence genomic in situ fluorescence genomic in situ hybridization applied to a chromosome squash preparation of a tetraploid. So this is tetraploid is going to have that hybridization event, right? That we just talked about. Amstoma lateral and Jeffersonium and barbori larva. from that forest. So alateral chromosomes are in green. Are they spatially, like, I'm thinking, are they, this is the one that has the multiple genome copies. So like, where are the genomes actually, the chromosomes actually stored? Are they separated? Are they spatially nearby one another? It doesn't seem like it. The A. jeffersonium chromosomes are in blue. Wow, they're everywhere. And A. barbori chromosomes are in pink. So I guess they're kind of like together. I wonder if there's like something like holding them together. The sphere at the bottom is the interphase nucleus, obviously, with a bunch of that. Can we see the different colors inside the nucleus? It looks like they can. That's a pretty image. That is very cool. A barbori is the 5th known species that can serve as a sperm donor to enable unisexual amstoma to persist. Wow. In keeping with unisexuals that use other sperm donors, A. barbori sperm is incorporated in unreduced unisexual eggs to elevate the ploidy to tetraploid and pentaploid levels. But unlike unisexuals that use other sperm donors, no triploids were found that have an A. barbori genome. Apparently, unisexual kleptogens steal A. barbori sperm from gynogenic reproduction and incidentally for ploidy elevation. What? That is so crazy. How do they? How do they even get the sperm? I guess they're having some type of like sexual reproduction with them, obviously, right? But why would they want to? Like, why would they? Usually, like, there's some type of like a physical barrier. When you see this with other insects and stuff, it's hard for them to actually mate with one another. You see it with like dragonflies and stuff like that, where it's hard for them to actually have their bits like touch and connect, right? But this is interesting. It just seems like they're very promiscuous and willing to have the sperm go everywhere. It must be, and honestly, because of that, you can actually kind of tell that they have these environments. They must be pretty nutrient rich, right? Because their kind of ability to create that sperm and release it to the unisexual kleptogens. That's really interesting because you'd think that there would be basically some evolutionary mechanism to stop this A. barbori sperm from going into another species or a unisexual kleptogen, right? You'd think that there would be some way that they would develop like some type of like, my phallus is a triangle and the opening is, I don't know, whatever is incompatible with the triangle. You know what I mean? So there is a lot of physical differences that usually happen. So this is like a really what is the evolutionary benefit for the A. barbori to have this breeding potential with this unisexual kleptogen? That's what I'm wondering. The Ohio population. So this Ohio population demonstrates the evolutionary significance and amazing flexibility of kleptogenesis. If one sperm donor is rendered unavailable through extinction or habitat alteration, another species can substitute as a sperm donor. So the geographical range of unisexuals is only limited by the range of acceptable sperm donors. As all unisexuals have a mitochondrial DNA that is most similar to A. barbori and very different from any other of the 4 sperm donors, we expected the genome replacement would be facilitated, and perhaps an A. barbori genome could even replace that lateral genome that is found in all the unisexuals. Wow. We found no evidence of that genome replacement. As most unisexuals have the LJJ genotype, we expected that the A. jeffersonian was present in the Ohio population, and that it's gone. All the unisexuals possessed an A lateral genome, which eliminates this possibility of these tetrads. Interesting. They're good. So they're going through the genome possibilities, the genome mating possibilities. Interesting. This is previous research on this. Plant flatworms that have this that had this done. Using microsatellites, they found that only 12% of the female's offspring were the result of partial genetic exchange that resulted in tetraploids derived from gynogenic triploid flatworms. So, wow, this is incredible ability to increase their genome size. They also, I wonder how different their environment is too, because normally like in the evolutionary mechanism for increasing genome size, how that actually translates is through a ability to deal with a lot of abiotic or biotic stresses. So abiotic is anything environmental, biotic is anything biological. So viruses, pathogens, that kind of stuff, predators. So I wonder if they just if that kind of stress, that selection is just the asexual reproduction strategy that they have just in general. These and other studies use molecular genomic techniques to improve our understanding of genetic variation in clonal female organisms and to document intergenomic interactions with males. It is evident that unisexual organisms need not be doomed by a lack of genetic variability. That is so cool. That is so cool. Mechanisms have independently evolved for female unisexuals to capture and use genomes from foreign donors. These facts defy any speculation or generalizations that unisexuals lack genetic variation and thus cannot evolutionarily compete with organisms that engage in normal bisexual reproduction. In addition to the obvious questions regarding the significance of sexual reproduction in assortative mating, unisexual organisms can provide answers to many fundamental questions in genetics, ecology, and evolution. The consequences of intergenomic interaction and the independent evolution of unisexual organisms are poorly understood phenomena. Yeah, why would this be the evolutionary answer? Why would this be because presumably this population of salamanders came from a speciation event, right? So there had to be some set of salamanders that actually did bisexual, reproduction and failed. The ones that did the unisexual reproduction they're the ones that survived. So maybe this is it. Maybe this is the way that they actually were able to survive, because they actually had more genetic complementation. They had more genetic variability. That's really interesting. They were able to increase their genome size really quickly. Wow. The consequences of intergenomic interaction and the independent evolution of unisexual organisms are poorly understood phenomena. Rather than being complex, unisexuals do not fit very comfortably with our expectations. The perceived genetic and evolutionary complexities associated with unisexuality are probably derived from inadequate study, but are more likely to be derived from the perception that normal bisexual reproduction drives evolution. Wow. That is so cool. Is that why there are so many distinct colors and sizes of dragonflies? So basically those bits that don't fit together are kind of usually that's for stuff that would normally be breeding together. So this is called cryptic species. So cryptic species are things that look identical, and before genetics we didn't know that there was two species there, but through genetics we can actually tell that okay, these are actually interbreeding populations that don't breed within one another. So they're different species, and it's there. There's the different colors and sizes of dragonflies is basically because of the evolutionary fitness of dragonflies and how strong they are in their environments. The males are passing on their genes, so what if it's not in their own species? The male sperm donors are not of their own species, and they contribute their DNA to it. So It's very interesting. It's a very, very interesting paper. I actually am kind of curious. We'll see. I'll do some more research. I'll see if this has been how you kind of like figure out if this has actually been cited by anything. I guess we can do it right now. I can kind of show what would you do. I'm really interested in this 2005. So this is going to be a paper that has been around for a while. So if people had wanted to cite it, they could cite it. So I went to scholar.google.com and now I found this paper, and now you go to Cited By. Cited By is a really cool tool because now we can see the experts in the field. What are they thinking about this? Do they think it's a cool idea, a cool paper, like I think it's a cool paper? So let's see. So we have some reviews: sex chromosomes in meiotic hemiclonal, clonal, polyploid hybrid vertebrates. Some reviews. Delete and survive. That's another review. Clonal polymorphism and high heterozygosity in the celibate genome of the Amazon molly. Celibate genome? Yeah, Titan. Yeah, exactly. So that's passed presumably to its own species and this other species. So it must have a lot of nutrients in order to like continually make this much sperm, right? Multi-scale chimerism. That is so cool. Okay, so there's a lot of like reviews for it. Doesn't seem like there's a lot of new research. Whoa, time and time again, unisexual salamanders are the oldest unisexual vertebrates. So maybe that's why we have these interesting new strategies, because they've kind of been around for a while. Very interesting. What was this paper? Clonal polymorphism and high heterozygosity in the celibate genome of the Amazon molly. Okay. We will take a look at this later. We'll save this. I'm going to tease it. I'm going to tease this. Celibate fish. How does it survive in nature? That is a cool thing to think about. Question from chat. Is it just that a green isn't interested in a blue, or is it that it physically can't even if it tries? Like literally the bits do not fit. They're incompatible. The cryptic species, like because they look so similar, you would actually have them breeding with one another, but you don't necessarily want that, right? For all the reasons why we talked about here, where it would be bad. The bits just wouldn't fit in the parts, you know. Cool. Very cool though. All right. Okay. Cool. So this is another one that we're going to go through. All right. So here we go. When did the dinosaurs go extinct? There's new science that are in the... One more time. That was sad. That was bad. And I need to do this anyway. All right, here we go. When did the dinosaurs go extinct? New fossil fish evidence is revealing that timing. Very cool stuff here. Let's think about that, right? The asteroid impacting the dinosaurs and making them go extinct. I have no idea when I have no idea. So winter began in spring for many of the animals during the final year of the age of the dinosaurs. Paleontologists studying fossilized fish suggest that the spring was in full bloom in the northern hemisphere when an asteroid slammed into Earth. triggering a devastating global winter and mass extinction. It's honestly, I can't even imagine that event, like the cataclysm that would have to be in order for something like that to happen. It's still like honestly boggles my mind. Like how could there not be things that lived, dinosaurs that lived like on the other side of the world or something, you know? But I mean, obviously they didn't. It's just, I guess you have such a cascading, bad event after event. And that's kind of the problem, that the entire globe just kind of crashed for these large. It's still crazy to think about, and it's crazy to think that we can actually know even what kind of time it was. So I wonder if this is the thing. Winter began, or the ability for spring to be kind of like there. Like I know we figured this out for the climate change. We figured this out by pollen. So we take the ice age, the ice caps, we drill into them, and then basically because of the hexagonal structure of water, when it freezes, has a little pocket of air that's inside of that hexagon, that 3D hexagon. All right. So this is the cool part. This is actually the reason why water floats when it freezes. You know, if you think about it, most things get denser when they freeze, right? Because you have the molecules coming together, actually coming like more closer together. Water molecules create that pocket in the center, and that actually allows it to flow. And that characteristic just in and of itself has kind of like transformed the world, right? So they use that little pocket inside to actually test the pollen, so they can actually tell what kind of plants are available, what was happening for the plant life. So then they can actually see what other things would actually eat and build their bones and live off of that kind of stuff. So it'd be interesting. I'm curious how they can actually figure out the exact time point here. Okay, but the conclusions are likely to stir controversy among some researchers, partially because the fossils come from Tanis, an extraordinary yet contentious geological site in North Dakota. What is Tanis? Some were puzzled by the results published in Nature, which are essentially identical to those of a separate paper published in Scientific Reports last December by another research group working at Tanis. Neither paper cites each other. Okay. So there we go. So this is the kind of the funny part. So some were puzzled by this. So they're basically not citing each other, and you have a little of a, you didn't analyze the data right. No, you didn't analyze the data right. I'm looking at stuff. Why didn't you talk about this? Very contentious. I wonder if there was like a problem kind of at the dig site or something, with these guys like getting into it. That's cool. I mean, this is when I say like, science is very contentious. Like people aren't willing to roll over and just kind of like give big breakthroughs away, right? They're very contentious with one another, and they're going to call people out if they see that there is something wrong. Then they're going to do that, and that's what's happening, I guess, right here. Pretty cool. Okay, let's go back here. News of Tanis's discovery first hit the headlines in the New Yorker magazine in 2019. The team behind the discovery, led by Robert D. Palma, now a PhD student at the University of Manchester, then published a description of the site in the Scientific Journal. What? Wait, the team behind the discovery led by this is a master's student? A master's student is leading a dig? I'm shocked. I don't know. I mean, maybe I'm really surprised about that, but maybe that's how it normally works. That's really interesting to me. Okay. They said Tanis captured what happened just minutes to hours after the asteroid struck Mexico's Yucatan Peninsula about 66 million years ago. Okay. According to the 2019 paper, the impact generated 10-meter tall waves in a shallow sea that stretched across what is now the southern and eastern United States. As one of these waves surged up a river valley in what is now North Dakota, it swept up the organisms in its path, along with plenty of mud and sand. That wave then retreated and dumped that material, forming the Tanis site. Man, a 30-foot wave isn't that big, though. Like, I know, like, a 30-foot wave isn't that big to be able to, like, I mean, no offense, but, like, I'm very, I don't know, dude. I mean, I've seen 20-foot waves in the ocean. They have a lot of energy, don't get me wrong, but, like, are they going to be going all the way up? I wonder how long they're talking about. Like, what is the actual distance? That is all. I don't know, man. This is really interesting to me. I don't get it. Okay, the research came under intense scrutiny because no other site on Earth is thought to preserve a detailed record of the day of impact. Dang, that's a really good point. But many researchers note that the 2019 paper did not include a detailed description of the site's geology, making it difficult to assess whether the geology can really be tied to the impact or another unknown catastrophic event that occurred perhaps thousands of years earlier. Interesting. Yeah, I thought you would have to have like a topographical map. You'd have to have something that you like really showed. Like, that just blows my mind. Okay. For a site of such potential importance, I'd really like to see a long format paper that dives deep into the sedimentology and stratigraphy of the site and supports it with lots of imagery and data, said Thomas Tobin, a geologist at the University of Alabama in Tuscaloosa. He says that this is particularly important given that there's a limited number of researchers to date that have the opportunity to visit Tanis. Why is Tanis so hard to get to? De Palma says that an upcoming study that will expand on the description of the site given in 2019, he also acknowledges there's a, quote, misconception that he is restricting access to the site. What? This De Palma guy, the research... What? But he insists that's not the case. In fact, the access has been more open now than the typical cases. What? One researcher who had access is Melanie During, who is now pursuing a PhD in paleontology at the... Man, I guess you do. You just go to a site and you just kind of like... That's where your PhD research is. That's kind of cool, actually. I mean, how would you know? But it's crazy. There is some predictive capabilities of science, so it's not insane to think that, like, okay, I could go to a region and just kind of like chip away and find something. Like, for example, I know that like science had the evolution of whales specifically. You know, obviously mammals evolved first on land, right? And then there had to have been that that event that they actually went from land to sea again in order to develop whales. So they, for whatever reason, the evolutionary kind of story led to like the west coast of India, I think. And then they went there and they dug and they found it. So it's like that, there's a lot of predictive qualities of science. So it's not insane that you would do that. I don't think that it's that crazy. I think archaeology would be a really cool thing to do. It's like you're actually like unmasking something that is completely unknown and never seen before, and you could actually be like a new species or a new family, maybe even. Yeah, paleontology too. Yeah, that's what I meant. I'm sorry. I archaeology is either of them would be cool. Okay. One researcher had access is Dylan. Her PhD is in Sweden. Okay. In August 2017, while she was at the Virgi University of Amsterdam. She had the opportunity to visit Tanis because of her instructor, geologist Jan Smit, had previously worked with Diploma at the site. It looks like a car wreck frozen in time. It's insane. There are fishes folded around tree branches. You can tell that this wave displaced everything. What? Okay, now I really want to see, like, what is this? They have to have pictures or something of this. There's no way. This Tanis site? We have to look for this in a second. Okay, the fossil record. During wondered whether a geological record formed on the day of the impact would preserve evidence that could pinpoint the event in a particular season. She says the growth patterns of certain fossilized fish bones at Tanis can reveal the season in which the fish died. Wow. So kind of like a tree ring kind of thing. You have like the ability to like see kind of like the growth period, I guess, maybe. I have no idea how you would do that. The bones grow rapidly in spring. Okay, the next sentence. The bones grow rapidly in spring when food is abundant, but slowly in the winter when food is scarce, creating a microscopic line of arrested growth in the bone tissue. Okay, so it is like a tree ring. That's cool. During collected fossilized fish from Tanis and then working with Schmidt and other colleagues produced high-resolution micro-computerized tomography models of 3 paddlefish jawbones and 3 sturgeon pectoral fin spines. The researchers say it was possible to identify lags, which are those tree rings that we can see in bones, in the 6 bones, and show that all 6 fish had died shortly after the beginning of a new growth period. So I guess they could have died during that particular event, or maybe one that happened the next year. So that would be like that'd be very rare though. That'd be very tough. This suggests their deaths, and by implication, the asteroid impact occurred during the northern hemisphere spring. During says the findings could offer clues about why the impact triggered extinction, wiped out some animals, including all non-avian dinosaurs, but not others. She speculates that the impact's timing might have been devastating for species in the northern hemisphere that had young to care for. There is also some evidence that southern hemisphere ecosystems recovered twice as fast after the extinction. What? Like, why would that be? Like, why would that be? Would that be from like biodiversity or something? Numbers? Oh, no, because of the location of it. So that makes sense. So if there's a secondary impact, obviously they're going to recover faster than the place where it actually happened. Wow. That's really, really interesting. What's up, guys? I think their argument about the season of death is convincing, says Michael Newerby, a biologist at Columbus State University in Georgia who is familiar with using lags to study fish. He says that he would like to have seen a larger sample size, but acknowledges the difficulty in assessing that. Yeah, how would you? You're like, keep digging. I'm not that convinced. You might have just one, man. You might just have one fossil that was preserved. But lags are contentious, says a postdoctoral researcher who understands bone analysis and the geology of the Tanis region. Dude, this person's going unnamed. They were asked by nature to give a response to this stuff. And they say, this research, it goes unnamed. That's insane. That's wild. The comment. That's wild. You'd think a violent event would guarantee the organisms wouldn't be preserved. Yeah. I mean, I can't. It's like hard to even imagine what's happening in these events, right? It's hard to even think about. There's going to be loss of predators. There's going to be loss. I mean, the biggest thing would be primary producers. So any type of as you go through the food chain, the first thing is the primary production. So that's anything that does photosynthesis. So grass, algae, cyanobacteria, all that kind of stuff. And then once you have those plants, die and stuff like that, then you have the herbivores die, and then it's over. So really, it's I would say the first thing to go. Although you might have predators would be a good thing, like, because just a decrease in number in general is going to cause them to kind of have those problems. But I would say the thing that would cause the most cataclysmic event would definitely be the primary production loss. It's crazy that this person that's normally in science, like, if you're talking scientifically, you're like, I'm going to bag on you, dude. It's just science. Like, don't take it personally. I've had lots of people kind of like, I've critiqued a lot of people's work and stuff, and you can kind of be like, sorry, man, like, I like you and stuff, but you just don't have the controls in order to say what this is. You know what I mean? Like, it's just, but they're in a comment for a piece, they're basically saying, but lags are contentious, says this postdoctoral researcher who understands bone analysis and the geology of the Tanis region. So they actually had some actual Tanis experience, which is crazy. They have requested anonymity given the controversy of the site. There is no uniform agreed upon definition of what a lag is or how you identify one. Wow. That is some shade. They say, and neither is there agreement on how often and why lags form. I thought we had just gone through that. Really? As such, the researcher questions the strength of the spring impact hypothesis. Double trouble? Several researchers have pointed out that the study is nearly identical to the study led by De Palma that was published the last year. This early study would also examine lags in fish fossils from Tanis and use them to link the event to spring or possibly summer. Adding to this bizarre overlap is the conspicuous absence of any mention of the De Palma et al. Paper by the During et al. Paper, the postdoc says. That is very suspicious, very weird. Yeah, it is De Palma. Yeah. During points out that her team's paper was submitted for publication before the De Palma team submitted theirs. So there might have been a scooping, like, you scooped us. They must hate each other, to be honest with you. Ours is the prior work and does not in any respect rest on the data or conclusions of the De Palma et al. Paper. She says, adding that her team therefore felt no need to cite the De Palma et al. Study, although the new paper acknowledges De Palma for providing guidance in the field and access to the specimens. Oh, that's funny. Similarities in the studies might have expected to be some extent when considering what the second study was born from the same site and used specimens on loan from our study area, says De Palma. He adds that the studies complement and independently reinforce each other. I can understand that point. I can understand that. I remember back when CRISPR was kind of like first coming on the scene, 2013, 2014, you had, it was unbelievable. The Church Lab, MIT, and then you had the Doudna Lab at Berkeley, just boom, 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 boom. Every month, Science Mag and Nature come out. So every month they had like rival papers in Science and Nature, and sometimes they would actually have double publications. Like, that's just crazy to me that, like, they would be making such big strides that they would be like, yeah, we're going to publish this, this finding and this finding. We're not going to combine them. We're actually going to just publish them together in the same issue. It was unheard of. So this is really interesting. If they find it like a hot thing, like one's trying to scoop one another, it's ruthless. We've gotten scooped. I've gotten scooped in my scientific career, and it sucks. It definitely sucks. It is one of those things where you feel like, and the paper, I'll be honest, the evidence that they presented wasn't the best too. So I felt really mad about it. I'll be honest with you. I felt really mad. So let's see what this is. Man. Whoa, this is just an editor's note here. 1 year after, straight up 1 year. Editor's note: readers are alerted that the Reliability of the data presented in this manuscript is currently in question. Oh my gosh. What? Appropriate editorial action will be taken once this matter is resolved. Dang. Let's see if that had it over here. Oh my gosh. They're probably so mad. It got retracted. No way. The scientific reports. Okay, that's there. Yeah, that's there. This during paper gone. Oh my god. They usually this might be a bad DOI or something, because normally if there's a retracted notice, it actually says it's retracted. So this is actually very weird. This is very weird. Okay, so it wasn't bad link. This is okay. Oh my god, that was close. But this doesn't have the editorial thing. And this does. Oh, there's another one in Egypt that has there's two of them. Here we go. Okay. Wow. But dude, they know to have this like hit, that is not good, dude. Look at the citations. 4 versus during 12. So that definitely is like, they're definitely, yeah, this one's popping off and this one isn't. Man. Oh, that is nuts. So these are the actual papers, right, that we're actually looking at, and these are those tree rings basically in the fish, looking for the growth marks. Like how they're using the same methodology, but one's being like saying that it's not reproducible and the other is. How does that? Oh, okay. So they're going the distance from the bone structure to the. That's weird. They definitely didn't do that here. They just like showed that they existed. Oh, I guess they do show that here. The carbon isotope along with the incremental growth. How is that? I mean, it looks very similar. From chat, Smithsonian quotes De Palma as saying there were multiple papers being worked on as a follow-up. Yeah, I mean, that's, if you have this happening, you're kind of like, your question, especially if you're trying to be in this field, it's kind of goes into question and creates a huge problem. So this is the NASA. Okay, so this is them, the Paleo, the site. Wow, this is a lot of things. That's pretty crazy. Fossil pterosaurs, which no other fossils exist at that time. Wow, that's huge. Drowned ant nests with ants inside and chambers filled with asteroid debris. Whoa. Dang. Tiny inhabited burrows for some of the first mammals in the area after the impact. Wow, so there really is something that kind of created like a stasis event. There has to be something. Wow. I want to see the actual site. Oh, so it's like it's like at the ends here. Wow. This is nuts. I had no idea. This impact, which struck the Gulf of Mexico, wiped out all non-avian dinosaurs and many other species. Like, that was it. So this is the crater, and that, dang, dude. So maybe it was 30 feet up there. Okay, so that makes more sense. So that it was 30 feet. They're approximating it's 30 feet all the way up in North Dakota with a Gulf impact. Okay, that's gigantic. Then, oh my gosh, I was thinking like the asteroid caused a 30 foot tall wave, and I'm like, that doesn't make much sense. That doesn't make any sense. Jeez. Oh, cool. There's a documentary on it, with the Attenborough documentary. I wish we could watch it. That'd be cool. All right. Okay. All right, guys. Well, thanks for being in chat. I see you guys. I appreciate it. All right. Oh, this is a bad one. All right, this will be a quick one. Okay. Okay, here we go. Yeah, this is the sad one. Sorry, guys. You gotta do it. Is woke beer sparkling water? If so, then yeah. No, yeah, it's just sparkling water from. Can you see it? Boom. Yeah, dude. I freaking love it. I've completely like dropped like beer and stuff, and I just drink sparkling water because I figured out that all I like is actually the carbonation and the coldness of the drink. I don't actually like anything else. So. Dang. So the unnamed there could actually be someone that was sent that paper in review. They could be like talking like, hey, I don't think so, bud. And they're like, oh man, so that's pretty nuts, man. That is pretty crazy though, to have that going on. I'm trying to. Yeah, dude, SodaStream is so good. That's like, I mean, it's basically the same thing. This is just has like a little bit of lime. That's not like the LaCroix crap that they do. It's like actually like has some flavor to it. But I just like the water, just like carbonated water. I'm like completely changed. I go to when I go out to eat or something. If they have like sparkling water, I do that. I like the soda water from Fountain Drinks now. Fountain drink water is so good. It's so carbonated. It's so crisp, and you like have it, and it's so good, man. I honestly love it. Okay. Okay, guys, I gotta cover this unfortunately, but it's gonna be it'll be a quick one. We'll be back on to the coolest things that happen in science, but we have to talk about stuff going extinct. Humans are negatively impacting the environment and causing a lot of species to go extinct that wouldn't normally be going extinct. We are changing their environments. We're changing and killing a lot of their food, their prey, the predators that are impacting the environments. And we're seeing this now in a lot of different ways. And one of them are especially larger mammals, larger animals. They're going extinct. So unfortunately, this dolphin population goes extinct after its last member gets entangled in fishing gear. Freshwater dolphin population has gone extinct in northeastern Cambodia after the last member got tangled in fishing gear and died. The Irrawaddy dolphin was found dead on a riverbank on February 15th. It was the last one of the particular stretch on the Mekong River in the Stung Treng province. Irrawaddy dolphins are found in the coastal areas, or were found, of South and Southeastern Asia. They only live in 3 rivers now. That's crazy. Myanmar, Indonesia, and Cambodia. The Mekong River that the Irrawaddy dolphins lived in was already scarce. The Mekong River is 2,700 miles long. It's the 12th longest river in the world and running from Cambodia to Myanmar. However, the dolphins only inhabit a 118-mile stretch. That is like nothing. That is so sad. The World Wildlife Fund estimated that there are only about 90 left in this area. The lonely dolphin had been monitored by experts. After noticing its injuries, they tried to work on it to save it. However, authorities said that they lacked equipment and catching techniques, meaning they were unable to catch the animal to treat it. Instead, they kept a close watch on the dolphin to make sure it did not get harassed by fishermen. Eventually, the dolphin died. Its injuries eventually prevented it from swimming well enough to hunt, so it died. A spokesperson for the volunteer-run campaign organization Blue Planet Society told Newsweek that Irrawaddy dolphins were not described as a separate species by science until 2005, when the world had already been on the brink of losing them. So this is those types of species where it's hard to see. Morphology alone isn't necessarily enough. We need genetics and stuff to see, and that those tools have only recently been able to come available to us. Overfishing, pollution, development, and habitat destruction is having a devastating impact on marine life. Unless we set aside more fully protected areas for our precious wildlife, we can expect this trend to continue, unfortunately. The main threat facing the freshwater dolphin is becoming a victim of the bycatch and the fishing gear. Fishing in the Stung Treng province is important. Every year they have a fishing holiday. Marine deaths from the entanglement fishing gear is a problem. An estimated 300,000 marine mammals die as a result of discarded fishing. Dang, man. That is insane. I want to look up the... I want to see the actual... Wikipedia for it real quick. Dude, what an awesome looking thing, man. That is awesome. Look at that picture. That is so chill. Oh man, the propellers. That is so cute. Is it gonna be? It has to be listed as extinct now, right? Wait, it's threatened? I'm so confused. Dolphin population goes extinct after... Oh, I think it's one of the members of the okay. So that's better. That's good. That's better news. So it went extinct in the it extinct of that river, but it didn't go fully. Okay, I'm understanding now what's going on. Dude, that is a cute dolphin. Yeah, we have to save this stuff. It's really important. Oh, okay. Yeah, a population of it, exactly. Yeah. I mean, I like personally, like, I think that we have to kind of focus more on ways that we can treat and we're like actually interacting with animals. It kills me that we are so flippant about this. These type of experiences and these type of things where we're actually interacting with wildlife can be completely life-changing for these organisms, or I'm sorry, for the humans that interact with them. So like for me, for example, like some of my favorite moments in surfing has actually been surfing with dolphins and having dolphins like underneath me and kind of like interacting with me And it was such a cool thing. It's such a bonding experience, like you with nature, bonding with the feeling so small in that moment, right? Feeling like, wow, these animals are so incredibly versatile, and they're so like they move really easily in the water, and I'm just flopping around, you know. It's pretty crazy. And like I think about that, and then I think about those intimate, incredible moments I've had with like those dolphins, where it's just like me and like 10 dolphins in the water, you know. It's really cool. You kind of like have this bonding experience with them. And then I think about things like the Steller sea cow, and how like the Steller sea cow went extinct, you know, in the same place in San Diego. When I'm surfing, I'm thinking like I got robbed of even when I'm snorkeling in San Diego. These used to be there. These basically they're manatees, but they lived in the Pacific Ocean, and they went extinct really quickly. I think it's like within 15 years of them being discovered or something by humans, they were actually went extinct, and they were hunted to, you know, because they're slow moving, they're big, you know. It was like one of those things where they're easy. And, yeah, I just think that they were so, I'm bummed, you know. I'm bummed that I actually can't. Oh, within 27 years of its discovery by Europeans, the slow-moving and easily caught mammal was hunted to extinction for its meat, fat, and hide. And it's just like, it's just not okay, you know? It's like, this is one of those reasons why we have to kind of, I mean, for a bunch of different reasons, for ecosystem services, you could think about it from the economy point of view, or you could think about it more personal view, you know, where it's like, you know, how are you going to get your food? You know, what bee is going to pollinate that plant in order for you to get your food? Or even more personal experiences like this, where it's like, if these things aren't out in nature, then I'm not going to be able to experience them, you know? And it's like, it just really kills me. And I think about, you know, I think about the moments that I was as a kid, you know, when I was interacting with, like, Catalina Island is off the coast of California, and I used to go there, and I used to, when I was a kid, run through like the little, like, not a forest, but like kind of like this little natural area and stuff, and we would run around, and, you know, it's just like we would catch crawdads and all this kind of stuff. And, you know, crawdads and shrimps are like some of the first things to go in polluted environments, you know. And like, I don't know, I probably would still be a scientist, you know, if I didn't have those experiences. But wow, what such a crazy, important, formative experience that I had, that where I really had this caring that was kind of established for the environment. And if we don't have nature for people to experience that, then, you know, what's going to happen? You know, what's going to happen to this stuff? So it just really saddens me. I'm happy that they didn't go fully extinct, but that it's not very good, you know, that at any point. Then we have to stop the overfishing. We really need to stop with the overfishing. From chat, I really would like to have seen the North American land sloth. That is exactly. Those things are so cool. They used to burrow into the ground, and they were giant, like the size of like polar bears. And yeah, those went extinct too. All the macrofauna, that's called macrofauna, is basically anything bigger than like a dog. It's all been going and it's going to be hunted to extinction, or it's being killed. Like, we don't want them. They're kind of like in our area kind of thing. So exactly, Nick. Yeah. All right. Anyway, I misread that title, but I'm happy that I was wrong there. That's good. Okay. What's next? Um, okay. Okay. So this is kind of like a pet issue that I have. This is kind of hard to. Wow. Okay, I'm just like going through here. Another one about extinction. A really, really soft topic. Well, this is a really cool paper, though. Okay. Some modeling. Okay, very cool. I have to be really careful. Okay, Nick, that sounds good, man. I appreciate you. I can understand. Okay. Can I like make this bigger? Oh, there it is. Whoa. I didn't want to make that bigger. That's annoying. Let's block this. Block. Okay. Okay. This is one of those things I have to be like careful how I talk about this issue because I can't downplay it, but I'm really frustrated at the framing that we kind of currently have in science. So I think it's a good topic to talk about. Okay. From chat, Doc Vierson. Crazy question. Is there any way humans could convert sunlight or other energy into food? Great question, man. I mean, are you talking about like as humans, like making humans able to do that? Because what you're talking about is basically photosynthesis, right? Photosynthesis is the ability and to create longer change of carbon from CO2 itself, and the energy from the sun is the thing that you use to do that. So there is, and if that's what you're talking about, this is kind of cool. So this is a I actually tweeted about this. I was like, it's so frustrating to me, the cool things that happen in science, and we just don't even like talk about them, or it's not even, people don't even it's crazy. Let me try to find it. Dang it. Dude. Sorry. This is really. I'm trying to find it on my Twitter. I know that I had a paper on it. Sorry, 1 second. I'm trying to find it. Let's see. We'll let biohackers try and figure that. Kind of like receptors, but maybe just rudimentary nutrients. Yes. See, this is the thing, dude. Like, I mean, this is my attempt to like get something that's like semi-viral or like hot, you know? Like, I'm bad at this stuff. I'm real bad. So I posted that because I'm like, dude, remember when science bioengineered photosynthetic pathways into mice and everyone just forgot about it? Like we did this. We actually allowed exactly what you're talking about. We actually put photosynthesis into science did, not me, but science put it into that system and incorporate it into the metabolic machinery of mice. So they actually did it, dude. Here's the paper. I'm so frustrated that this isn't getting the I think it's on here. It's on my my stream agenda. I just haven't. Nope, it's not actually. I'll put it on there. I'll make a video about it. I know, dude. I know. I mean, it's like this. This is the whole point of my channel. My science channel is trying to just we have in my mind. I have like absolute banger after absolute banger of content. You know, these crazy papers where you're just like, how this should like stop the presses and everyone just kind of like everyone should be focusing on it. Oh, thank you for that. I finished my PhD, so that's the kind of break for the last 6 months that I had. I kind of had to focus on that, write my my thesis. I had to defend my thesis, all that kind of stuff. So I got a discussion, a debate that I did with LectureFan up today, but I'm going to be putting more time into the channel just in general. Because I mean, obviously I have a lot of fun like going over this. How could you not, right? It's just insanely cool, the amount of stuff that's happening in science. And there's just an endless supply of it. And it's just so cool. So, oh, thank you. I appreciate that. So, I mean, this stuff is so cool. They actually incorporated it into ATP synthase. Thank you. It's like I can't even grasp like how freaking cool this is. It blows my mind to actually put it into the TCA cycle and stuff is just so cool. I was planning to do 1 year in industry, then maybe attempt my PhD. Yeah, this is one of the topic ideas that I have. Kind of like going to grad school. I'm going to do this right now. I'm a professor. Thank you. I'm a professor in microbiology right now, and I'm kind of giving a lecture that I wish I kind of got coming out of undergrad, and I'm going to have the slides and I'm going to kind of prepare it, and I'm going to like basically do it for like a video on YouTube. Any of my lectures I can't put onto YouTube, but I can actually I can use that material and stuff. But basically what I recommend is not going straight to grad school out of undergrad. I just don't. There's just too many things. There's too many considerations, and your life is too heavily impacted to just go from school, big chunk of school, and the problem is that you really don't know what you want to do your PhD in. Maybe you have a inclination, maybe you have like, oh, I think that this stuff is cool, but like that's a lot different than like actually doing the science. Like, for example, like, oh, I think genetics is interesting. I like that type of, compare and contrast and all that kind of stuff, and seeing how like hierarchical charts will happen. And that's kind of like how it's classically presented. But modern day genetics, actually doing genetics, is computer science actually. So you're using computers and, Python or, a Perl to actually align millions of sequences of DNA and try to figure out if there's a single nucleotide change there. So you really have to like work in the field that you are wanting to work in first. So that's why industry is so good. So then you can kind of be like, is neuroscience for me? I can work on a pharmacology company and actually work with that type of applications. And then you're like, okay, this is good. And then you can go put your PhD in there. Yeah, exactly. You want to have some experience. You want some type of experience in the field that you're working in, because otherwise those are those hell, you have an ability to rotate once you're at a PhD program. Normally you have to check this actually. Some grad programs don't allow you to rotate, and I would recommend rotating for sure. So make sure you have your labs and your rotation figured out, and you love the labs, because you want to have that relationship with you and your PI pretty good. So, but yeah, I'm going to like flesh this out more, I think, through like a bigger video, and kind of like go through the steps of like why you'd want, coming out of undergrad, what you kind of want to do. You basically have a biotech or an industry job. That's what the people call it. Or you could work at like a national lab, or you could go straight into school again. So that would be like a graduate program, veterinary school, medical school, anything like that. No problem, Doc. So anyway, cool though. Nice. Yeah. I mean, that's basically what me and my wife did, where working at a biotech, I was able to basically pay for the both of us to live. And then she was basically able to pay for both of us to live while I was at grad school. So it was a huge win for the both of us, because we kind of both got that ability to focus on education. And that was really nice. So it was very nice. Grad school in general, I had a great time. I thought it was a good experience. You just have to know and kind of get out of it the most you can. And a lot of people that don't, it can be a hellish experience. Policy or government jobs are an under pursued pathway too. Yeah. So yeah, science policy, that's an interesting, let's see. Like, how would I want to talk about that? I would want to say, I'm starting to take notes here. Out of grad school, you can do undergrad, grad school, national lab, industry. So science at a policy thing. So basically what you're doing there is, so you have lawmakers that need questions answered about a variety of topics. So what they'll do is they'll basically say, okay, I have a law that's going to be passed on vaping. Okay. So I need you to collect all the data that is surrounding vaping and data. So you're going through and you're saying, okay, there's evidence to say this, there's like the evidence to say this, and you're reading a bunch of literature reviews, and then you're making that into a 10-page paper, the review basically, and then you're giving that to your congressman, and maybe you have an abstract that's like, smaller, maybe a couple hundred words, and then you have like an executive summary that's like maybe a sentence, and then maybe they read that sentence. So it's not like, it's a very thankless job, and whoever you're in, the congressperson that you're in, is really gonna be very varied, your experience. So like, how you don't really do science for lawmakers, you advise them on the science that's already been done at national labs or universities, and then you portray that. Government labs or national labs are really cool and really underutilized in general, just for spots that you could go to. And I would recommend, some of the coolest science that's happening is at national labs. Yeah, honestly, it's one of those things where I actually, if I was given the choice, I would probably actually go to like a Republican lawmakers if I could, just to kind of like give them that ability to to push back and be like, this is what the evidence shows. This is what's happening. I don't think you could request party affiliation. That you just kind of assigned the AAAS. This is what it is. So the AAAS puts it on. It's the Science and Technology Policy Fellowships, and I've known people that have done this. I mean, it's one of those things where, oh, I actually know this guy. This is who I know. That's funny. Yeah, national labs are really cool, especially the Department of Defense. I know people kind of hate that idea, but some of the coolest out there research is actually happening at national DoD labs, because they want to do the crazy out there stuff, and that's like some of the coolest stuff to the most fun science for me to do. That's, so like, for example, my like delivering DNA to chloroplasts of plants for vaccine production. The NSF and the DoD also, all checked it out. But you're gonna like that those labs and stuff. You're gonna get, there's basically at biotechs and national labs and all that kind of stuff. The good point is that there's all levels of people, right? There's all education levels. So don't just think like, oh, I don't have a PhD, I can't do this, or I actually spent 5 years in industry. I didn't want to spend 5 years. I was applying for grad school for 3 years of those years. So I was kind of like hoping to get out of industry, and they grad school wouldn't take me. So so it was kind of crazy. I actually failed to get accepted into a program for 3 years. 35 programs denied me. And but the third year, my wife and I ran out of money. So we had to go, and we had a car that we owned. So we had to go to a bank and, as collateral, take out a car loan on that car so that they could give us, like, I think it was like $7,000 or something. So then we could actually use that money to apply to grad school. So it was, graduating this year was nuts, because it was, it's a 9-year process between the 3 years that I was applying, then 6 years at grad school, and the pandemic and all that kind of stuff that kind of extended me another year. So it took me 9 years to graduate with my PhD. So that was, it was tough. I'll be honest with you. It was very tough. Yeah. So what you could do with the kind of like the really cool idea is actually to kind of like apply to the NSF, apply to national, you can apply here. I'll give you the link. So like the NSF GRFP is a program that you can apply to, and it's crazy. You can actually get money from the NSF, and then you could walk into like any like grad program that you wanted. So if you had like a background, this is the route that I wish that I would have done, is basically gone and gotten an NSF GRFP, which I was awarded. After all of those rejections and stuff, I actually was awarded one of the prestigious research fellowships, and then you could go anywhere. Any university is going to take you because you're funded. You're fully funded. So you could just go into any university, at any professor is going to want to take you, and you just freaking like, hey, guess what? Like, I got NSF GRFP. Guess what I'm doing? So I wouldn't be nervous, man. I would think like, you want to do something and you're not, like grad school isn't the place to figure out whether or not you like science or not. I'll tell you that. It's a place, and academia in general is a place there you need to like push yourself to take advantage of every opportunity that you have. If you're not willing to do that, then you're probably in the wrong spot, and your time would be better spent at like a biotech or something like that, because you can become a scientist at a biotech too. Scientists like work at biotechs as well. Don't listen to those academics that are like, oh, you can only be an actual scientist with hypothesis driven research at an academic institution. Like, that's BS, dude. Don't listen to them. So that way of thinking, that professor old hat kind of thing. Anyway, okay, this is a big one. This is like oh, this is a big paper. I wonder if I'm going to do something else, just because I want to give this like a really good scrub. It's hard to explain. Basically, I have a running debate with my science friends, like what's the major contribution to mammalian, the giant extinction event that's happening right now. And I think that basically what we're talking about CO2 too much, and what we need to be talking about is like human activity, humans actually like living in these places, destroying habitats and all that kind of stuff. So this is kind of like a paper that's kind of interesting that is showing that. But I need to be careful on how because I'm going to have my scientist friends really scrutinizing everything. So I might save this for another time. Okay. Okay. Have a good one, man. Adios. Thanks for coming. Thanks for watching the stream. I appreciate you. All right. I'm going to do one more. Because I still got to make thumbnails and all that kind of stuff. I'm looking to hire an editor, trying to make this process a little easier. Because I like talking about this. I like reading the papers. I like talking to and answering questions about science, but I don't necessarily like the thumbnails game with YouTube and gaming the algorithm and the editing and all that kind of stuff. So I kind of want to hire an editor if I can. That'd be very nice. Okay. Let's do one more paper. And then that's a significant chunk of the backlog chewed through. Okay. This is a cool one. And it's about algae. So everyone, this is, I actually got, based off of Levelhead, I actually got, because he calls me the Algae Christ, so I actually got the algaechrist.com. It's actually my URL now. There you go. Pretty funny. Oh my god, the URL's not working. There it is. Okay. Pretty funny. Okay. All right. So let's talk about this. I'm trying to find this paper. Where is it? Here we go. No, I don't have the full. Oh my god, I don't have the actual. Is this real life? I don't have the actual publication to look at because it's behind the paywall. Let me see if I can get it. Damn. That's funny. Let's see if we can go through Sci-Hub. Sci-Hub. Rescue me. Let's see. Doing totally okay things here. Oh my gosh, the Sci-Hub doesn't have it. Okay. All right, guys. I think that might be, or I could do one more maybe. 1, 2, there's four videos. I think I need to do 3. So fourth one is kind of good. And I have the debate with Fabian Liberty. I have to do a whole COVID origins like expose, kind of like talking about it. So I have all these papers. So the big thing, and again, I'm actually giving I'm a professor of microbiology right now at University of California, Riverside. So I'm actually giving a presentation, a lecture on SARS-CoV-2, and because there isn't one. How crazy is that? How could you, like, just coming out of a pandemic, still in a pandemic technically, like, how are we not covering SARS-CoV-2, in a microbiology class? So I feel like I have to. And so I need to read all of these papers and go through them. And I want to present it. I want to present this stuff and go through all of the actual evidence and stuff for the origins of SARS-CoV-2. And I think it'd be a really cool stream. I think it's going to be a really cool lecture that I'm going to give. I'm excited about it. And also I'm excited to go through it. But not tonight. Not tonight. So I'm going to try to do like one stream a week. I think I'm going to try Wednesday works for me. Wednesday's kind of nice. I don't really usually have anything going on Thursday mornings. So that's kind of nice. So let's make a date for Wednesday. Let's do Wednesday, next Wednesday. Does that sound good? Good? All right. Thank you, everyone, for coming. Thank you. You're far too kind. I appreciate you watching, all the chatters. I appreciate that too. It's really nice to not have streamed in a while and then to have people coming in. I appreciate that. But that's it. I'm gonna bow out now and go forward and make some videos. Ministry of Science is going to be on Tuesday, and I am going to release the videos that we covered here. And I think next week we do the big one, the SARS-CoV origins, all the evidence of it, the scientific evidence of it. You've heard about the bad evidence, the non-evidence evidence, but how about actual evidence? I think you're going to be impressed and surprised by the amount of evidence that we have to actually talk about and elucidate actually where SARS-CoV-2 came from. But not tonight. All right, everyone. Vaccines are safe and effective, and may all your pipettes be accurate and your R-squared values be strong. Thank you.